Have you ever wondered how a scientist goes from just a simple question, like why do leaves change color, to a huge, groundbreaking discovery? Well, it's not magic, it's a method. And in this explainer, we're going to break down that exact problem-solving playbook that biologists use to unravel some of the planet's most complex living puzzles. So here's our roadmap. We're going to follow the entire journey step by step. We'll kick things off with the basic toolkit, then learn how to gather clues, form an educated guess, put that guess to the test, and finally see how all that new knowledge gets shared with the entire world.
All right, let's start at the very beginning with the foundation. What exactly is this powerful tool that sits right at the heart of all biological research?
Well, it's called the biological method, and a great way to think of it is like a recipe for discovery. It's the structured process that takes a scientist from a place of not knowing to a place of understanding, and it does it all by using hard evidence as its guide. As you can see, this method follows a really clear six-step path. It all starts by just recognizing there's a problem. From there, you make observations, form a hypothesis, test that idea, and finally share your results. So let's start at the beginning, gathering the clues. That's right, pretty much every scientific journey kicks off with simple, pure curiosity. This whole first phase is all about asking questions and gathering those initial clues.
So step one is just recognizing that a biological problem even exists and then framing it as a question. It could be something as huge and as critical as asking, what are the risk factors for lung cancer? That one question defines the entire investigation that's going to follow.
So, once that question is set, the biologist basically puts on their detective hat. The next step is making observations, and that means using all five senses to gather raw data and information about the problem in front of them. Now here's what's really interesting. Observations break down into two main types. You've got qualitative observations, which are descriptive, like the color or the texture of a plant. But then you have quantitative observations, and these deal with numbers, like its height in centimeters or its exact temperature. In science, that quantitative data is usually considered more accurate, and that's because it's measurable, it's objective, and you can record it precisely.
Okay, so you've asked a question and you've made your observations. What's next? Well, now it's time to form an educated guess, a potential answer to your question. And this potential answer has a name. It's called a hypothesis. Now, it's really crucial to understand this is not just some wild guess. It's a tentative, informed statement that comes directly from the observations you just made. It's your first real shot at trying to explain the why behind the what.
But, not just any statement can be a good hypothesis. It has to check four really important boxes. It has to be based on observation, it has to propose an answer to the problem, it has to be testable, and this one is maybe the most important, it has to be falsifiable. That just means there has to be a way to prove it wrong. If you can't possibly disprove it, it's not actually science. And that brings us to the most critical phase of the whole biological method, putting that educated guess to the test. First with logic, and then with a real-world experiment. So before a biologist even steps foot in a lab, they make deductions. These are just logical predictions that are based on the hypothesis, and they're usually framed as an if-then statement. For example, if my hypothesis is true, then this is what I expect to see happen. It's this if-then logic that really shapes how the whole experiment gets designed. And then comes the experiment. You could call it the moment of truth. This is where you actually test your logical deductions against reality. The entire point of an experiment is to see if the hypothesis holds up under pressure or if it needs to be thrown out. This is where ideas collide with hard evidence. So a proper experiment always needs a comparison. The experimental group is the one where you change a single thing. For instance, a plant that gets no carbon dioxide. The control group, on the other hand, is kept under the exact same conditions, except for that one thing you're testing. So that's a plant that does get carbon dioxide. By comparing the two, you can figure out the effect of that one specific condition. All right, the experiment is done, the data is in, but the work isn't over. Not even close. Now comes the final and really crucial step, making sense of it all and sharing what you've learned. A biologist analyzes all that data to draw a conclusion. Was the hypothesis supported or not? But the science doesn't just stop in some lonely lab. The findings get published in journals and presented at conferences. And this is absolutely vital because it allows other scientists to check the results, to build on that work, and to use that new knowledge to solve even more problems. In a very real way, science is a team sport. So, why does this methodical process matter so much in the real world?
Well, let's ground this with a really powerful historical example. More than 2,000 years ago, physicians knew about a terrible illness they called the disease of chills and fevers. We now know it as malaria.
And the stakes here could not have been higher. Over the long course of human history, malaria has killed more people than any other single disease. It was a devastating biological problem that absolutely demanded a solution that could only be found using this very method we've been talking about.
Our understanding of malaria, the development of vaccines, breakthroughs in genetics, all of it is built on this exact methodical approach. It really is humanity's most powerful tool for turning curiosity into knowledge and then turning that knowledge into action. Which, of course, leaves you with one big question. How many other major challenges have been solved and will be solved using this very same process?